This video tutorial is about the need for cellular respiration. Why do living organisms need to respire? Respiration is the process that occurs in living cells and releases the energy stored in organic molecules such as glucose. The energy is immediately used to synthesize molecules of ATP from ADP and inorganic phosphate. ATP in cells can be hydrolyzed to release energy needed to drive biological processes. Microorganisms, both eukaryotic microbes such as yeast and prokaryotics such as bacteria, as well as plants, animals, fungi, and protoctists, all respire to obtain energy. Energy is the capacity to do work. The energy that is stored in complex organic molecules, so fats and carbohydrates and proteins, is potential energy. It is also chemical energy, converted from light energy during the process of photosynthesis. When this energy is released from organic molecules via respiration, it can be used to make ATP to drive a whole range of biological processes, such as active transport, endocytosis, exocytosis, synthesis of large molecules such as proteins, DNA replication, cell division, movement, activation of chemicals. All the chemical reactions that take place within living cells are known collaboratively as metabolism or metabolic reactions. Anabolic reactions are metabolic reactions where large molecules are synthesized from small molecules. Catabolic reactions are metabolic reactions involving the hydrolysis of large molecules into smaller ones. Within living cells, atoms, ions, and molecules have kinetic energy, and this allows them to move. For example, when molecules diffuse down a concentration gradient, moving from one place to another, they use kinetic energy to do so. The diagram on the slide in front of you shows the energy transfer between and within living organisms. We're now going to consider the role of ATP. ATP is the standard intermediary between energy releasing and energy consuming metabolic reactions in both eukaryotes and prokaryotes. This image on the slide in front of you shows the structure of an ATP molecule. It is a phosphorylated nucleotide. Each molecule of ATP consists of adenosine, which is the nitrogenous base adenine plus the 5-carbon sugar ribose and 3 phosphates or phosphyl groups. ATP is relatively stable, i.e. it does not break down to ADP and P when in solution in cells, but is readily hydrolyzed by enzyme catalysis. However, whilst in solution, it can be easily moved from place to place within a cell. The energy-releasing hydrolysis of ATP is coupled with an energy-consuming metabolic reaction. ATP is the immediate energy source for this metabolic reaction. When ATP is hydrolyzed to ADP and P, a small quantity of energy is released for use in the cell. Cells can therefore obtain the energy they need for a process in small, manageable amounts that will not cause damage or be wasteful. ATP is referred to as the universal energy currency, as it occurs in all living cells and is the source of energy that can be used by cells in small amounts. Some energy is released from the hydrolysis of ATP as heat. The release of heat, both in respiration and during ATP hydrolysis, may appear to be inefficient and wasteful. Heat, however, helps keep organisms warm and enables their enzyme-catalyzed reactions to proceed at a near optimum rate.